Good day, everyone, and we are now in our second lesson for immunology and serology, and this is about the nature of antigens and the major histocompatibility complex. Before anything else, I am Dr. Subhachay Basit, your lecturer for this particular lesson. So, are you ready to learn? Let's go! Okay, so for this particular lesson, we shall be discussing what are the different factors that may influence the immune response, what are the different characteristics of immunogens, the traits for that matter, and then we shall be discussing some of the important terms in order for us to understand this lesson further, such as epitopes, haptens, and adjuvants, then the relationships of antigen to the host, the major histocompatibility complex, the MHC, and its role in the immune response. Okay, so for the first lesson, we will now be discussing what are the factors that may influence the immune response. Okay, first is the age. So remember, okay, the older the individuals, okay, this will decrease response to antigenic stimulation. Okay. And then if a child is a newborn or a neonate, the immune system is not yet fully developed. That's the reason why when my son was born, okay, so when my son was born, he received BCG, then hepatitis B vaccine. So he did not receive the entire 6-in-1, 5-in-1, pneumococcal, not yet, because at that time his immune system is not yet fully developed. Okay. And then, of course, the overall health of that individual. Okay, if you are properly nourished, that's good. But if you're undernourished, then your immunity may be affected as well. And if you are fatigued or stressed, this may also affect your immune system or the immune response. Okay, the route of inoculation. When you say route of inoculation, it depends on how you'll be receiving, let's say, for example, vaccine or the antigens. So, of course, the ideal way of receiving it would be by means of subcutaneous, intradermal. Some may be intravenous. However, there are some vaccines that can be received orally. Okay, such as the savin vaccines for the oral polio, and the other one is the vaccines for the rotavirus. Okay, so they are actually being received orally. What's the reason for that? If you remember our previous lesson, the reason is that there is a MALT, mucosa-associated lymphoid tissue, and this particular MALT should allow antigenic will be stimulated by the oral vaccines that you will be ingesting, and this will stimulate mucosal immunity. Okay, dose. Of course, the dosage for infant and for adult will be entirely different. So it says here, the larger the amount of immunogen exposed to, the greater the immune response. Okay, however, you do not want an excessive dosage of a particular antigen because this will induce immunologic tolerance, which means that you'll not be able to stimulate an immune response at all. Okay, so the genetic capacity or predisposition of a certain individual. So it is actually unique from one individual to another. Okay, and what should be the important traits of immunogen? Immunogenicity depends on different factors. One of the most important factor is its molecular weight. Okay, the macromolecular size. Okay, the minimum requirement should be 10,000 Daltons to be recognized by the immune system, and most active are at least 100,000 Daltons. So any molecule that is less than 10,000 Daltons are not considered to be as an antigen, so it is called hapten. Okay, foreignness. The more taxonomically different it is, the more successful it is as an immunogen. Okay, so that is the reason why an organ transplant between human and monkey, we call that xenograft. There has been no documented proof that it's gonna be successful. Okay, so what is an epitope? The epitope is defined as the site of antigenic determinant. Okay, so that's our basic definition of an epitope. So a certain antigen would have a site of antigenic determinant, and that is called epitope. It is the key portion of the immunogen recognized in the immune response. By the way, I've been using the term antigen and immunogen interchangeably, but there's actually a contextual meaning behind it. So when you say antigen, it is something that can stimulate antibody generator, which we had previously defined in the previous chapter. But when you say immunogen, immunogen is an antigen that is capable of eliciting an immune response. Okay? So an epitope, therefore, is a key portion of the immunogen recognized in the immune response. So there are several forms of an epitope. So an example here is the linear epitopes, wherein the amino acids are sequential, which means that amino acids following one another on a single chain. That's why it is being called linear epitope. So there is also what you call a conformational epitope. As you can see, has more than one, has folding of one more chain. So you have a folding here, and then you have another folding here. So what's the advantage of a conformational epitope? It may allow amino acid from different segments to come into close proximity. Okay, which means that somehow antibody attachment for this particular epitope will be much easier. Let's say, for example, if the antibody is supposed to bind at green antibody, and if it's a 2-valent antibody such as an IgG, so it can bind here and bind here. Okay, so that's conformational epitope. So I told you a while ago, if the antigen is less than 10,000 Daltons, it is not supposed to be called as an antigen, but instead we call it as an hapten. So what's the difference between a hapten and antigen? Antigen is supposed to be immunogenic, while hapten is non-immunogenic unless if it's combined with a carrier. Okay, so haptens are non-immunogenic materials that create new antigenic determinants when and if only combined with a carrier. Okay, so it can react with the antibody even without being complex to a carrier molecule. Yes, it can react, but it is non-immunogenic if it doesn't have any carrier molecule. Okay, so when bound to carriers, okay, it contributes to the development of interconnected lattice. Okay, the structure that you are able to see here, we call it lattice formation. Okay, why is it important? Lattice formation means that the antigen and the antibodies have already been complex in a way, and this particular complex antigen and antibody will allow us to observe precipitation and agglutination reaction, which we will be discussing in our future discussions. Okay, so adjuvants. What are adjuvants? Adjuvants are actually substances that will render vaccination more efficient. Okay, so it can make immunization more effective. And adjuvants primarily work by targeting APCs. You remember APCs? Macrophages, dendritic cells, mast cells. Okay, APCs are antigen-presenting cells, important to the adaptive immune response. Okay, it protects immunogens from degradations. So it makes the vaccine more stable, and it allows a longer response time that attracts more immune cells to injection sites. So immune response would be magnified if adjuvants are present. So you also use to accelerate, meaning it makes the process in a much faster, okay, and increases duration of protective immunity. Okay, aluminum oxide is an example of adjuvant. Okay, so let's talk about the relationship of antigen to the host. Okay, so autoantigen is something that a misnomer, I should say, because when you say antigen, it is foreign, but if it's auto, it means yours. So it's self. So autoantigen is like self than self. Anyway, so when you say autoantigen, these autoantigens belong to the host. They do not usually evoke an immune response. And if immune response occurs, autoimmune condition is most likely. So autoantigen, therefore, are actually one of the reasons why autoimmune diseases could possibly be present. Okay, alloantigens from other members of host species, such as in transplantation and blood transfusion. So we call it the alloantigens. Okay, so for example, you are a patient and you need blood donor from other person. So same species, Okay, so we call it alloantigen. So the blood that is being given to you, that's an alloantigen. And then the kidneys coming from other people, then you need it. So it's being grafted into your body. So that kidney is known as the alloantigen. Okay, and heteroantigen came from other species such as animal, plants, or microorganisms. So SARS-CoV-2, heteroantigen. Dust mite, heteroantigen. Pollens, heteroantigen. Poison ivy, heteroantigen. Food, heteroantigen. Shrimp, because some of us may be allergic to shrimp. Okay, heterophile antigens. Okay, so this is quite complicated. So when you say heterophile antigen, it exists in plants or animals, but identical or closely related in structure, so that antibody will somehow cross-react with antigen of the other. Example, so if you get infected with Streptococcus pneumoniae, that's a pneumococcal bacteria, so the antigen coming from pneumococcal bacteria will cross-react with those people who are type B. Because you're type B, you have a type B blood. Okay, you have a B antigen on the surface of your RBC. So what's the complication here? The complication here is during, for example, cross-matching. So a cross reaction may not be attributed to mismatched blood type, but somehow because the patient had been infected with pneumococcal bacteria, so this is where confusion happens. Okay, so there, heteroantigen, heterophile antigen, and in the future we'll be also discussing heterophile antibodies, but entirely different topic. Okay, now let's talk about the major histocompatibility complex. So the major histocompatibility complex is linked to the genetic capability that allows the body to mount an immune response. Okay, so it varies from one individual to another. So this explains the reason why some of us are more predisposed to have rheumatoid arthritis than the other people. Okay, or why some of us are allergic to shrimp. I love shrimp, but some of us are actually not. Okay, so genetic predisposition is actually the reason for that. Okay, so it have been found in all nucleated cells in the body. Okay, so the major histocompatibility complex are found in all nucleated cells in the body. So later on we will be discussing the MHC class 1 and MHC class 2, and it plays a pivotal role in the development of humoral and cellular immunity. So what's the application of the major histocompatibility complex? Because each of us have different set of MHC, 3 coming from our mom and the other 3 coming from our dad. So this may somehow determine whether the transplanted tissue is histocompatible, accepted, or recognized, depending on the percentage of identity. Okay, so theoretically speaking, if you have twins, you will not have any problem because you have the same genetic makeup. So the chances of you rejecting the kidneys of your twins are least likely as compared to if you're getting kidneys from a random individuals, because you are you as a person, as a recipient, and that random individual as a donor may have a different MHC makeup. Again, 3 coming from your mom, another 3 coming from your father. Okay, so major histocompatibility complex molecules will bring antigen in the body to the surface of the cells for recognition by T cells. Okay, and when combined with antigen on the surface of the other T cells, it will activate the T cells. So, what are the genes coding for MHC molecules? So take note that the genes that encodes for MHC molecules are found the short arm of chromosome number 6. So again, as what I've told you, there are actually 3 classes of MHC molecules. So what are these 3 classes? So class 1 is found in different loci. So it is designated as class A, B, and C. Class 2 is actually found in the D region, and this is designated as DR, DQ, and DP. And class 3 are actually complement components. Okay, so class 1 and class 2 are involved in antigen recognition, meaning to say, they influence the antigen to which T cells will respond to. So that's class 1 and class 2. Class 3 is composed of secreted proteins that have an immune function, but they are not expressed on cell surface. So the one that are found and on nucleated cells are classes 1 and 2. Okay? So alleles, okay, are alternate forms. If you remember cytogenetics, we have the term alleles are alternate forms of a gene that code for a slightly different variety of the same product. Okay? And HLA has over 2,000 different alleles. HLA-B has over 2,600 different alleles. C has over 1,500 different alleles. Okay, so it's quite difficult to get kidney donors that would have the same genetic makeup as you are, because not everyone has twins, right? So when you need kidneys, most likely you'll be getting it from your within your family. But even if it's your sister or your own brother or even your mother or father, your genetic makeup is entirely different. Okay, so that's the reason why most of these kidney transplant recipients would need to drink immunosuppressive drugs so that they will not be rejecting kidneys coming from other people, but it will make them immunocompromised. Okay, so again, it is being dictated in chromosome number 6. Okay, for the class 1, class 2, and class 3. Class 1 has HLA-A, B, C. Class 2 has HLA-DR, DP, and because the MHC genes are closely linked, inheritance for the grouping is known as haplotype. Okay, haplotype. So, one haplotype is inherited from each parent. So, again, so if you're able to see this one, each individual has 2 haplotypes. So, and then, so 2 haplotypes. Why 2 haplotypes? Because one haplotype come from your father and the other haplotype come from your mother. So for example, A1, B2, C3 came from your father, A1, B4, and C2 came from your mother. Okay? So both alleles make up the genotype. Okay? So if these are your haplotypes, your genotypes are A1, B2, B4. Okay? So the major histocompatibility complex have molecules on the plasma membrane. Okay, so we call it human tissue cells have molecules on the plasma membrane, and we call it the MHC. So these cell surface proteins are called HLA in humans. So again, as what I've told you in the previous slides, they are important in antigen presentation and even in the regulation of the immune response. So because of the differences in our HLA, these are the reasons why graft rejection during organ transplantation is actually taken place. So let's discuss MHC class 1. So take note, MHC class 1 is consists of a single chain. There you have it. Single chain anchored into the cell membrane. So if this is the cell membrane, okay, and then if this is the MHC class 1, you would notice that it is class 1 because it has a single chain, and this one is being stabilized by beta 2 microglobulin. So the molecule forms a group. So here's the group which holds antigen peptides, and it will present antigen to cytotoxic T cells. Where can we find MHC class 1? MHC class 1 is found in all nucleated cells of our body. So it will present to cytotoxic T cells, which are the CD8. MHC class 2 are made up of 2 chains. Again, class 2 chains. Okay? And class 2 present antigen to T helper cells. Okay? Kanina class 1 cytotoxic T cells, in class 2 T helper cells, or the CD4. Cytotoxic T cells is CD8, T helper cells CD4. So the groove, here's the groove, allow it present to larger peptides than class 1 molecules. Okay, where can we find class 2 molecules? Class 2 molecules are found in antigen presenting cells. Okay, so it will present antigen to helper T cells. Okay, so yan, class 1 is present on nucleated cells. It will, let me go back to that slide. Okay, so class 1 is present on all nucleated cells. Okay, class 2 is present on antigen presenting cells. Class 1 will actually present to CD8 or cytotoxic T cells. Class 2 will present to CD4 cells. So what are examples of antigen presenting cells? So these are macrophages, B cells, dendritic cells, mast cells. Class 1, the HLA are known as the A, B, and C. Class 2, these are the DP, DQ, and DR. Okay, so as previously discussed, class 1 molecules are expressed in all nucleated cells, and they differ in level of expression, which means that the class 1 MHC molecules are highest on lymphocytes and myeloid cells, lowest or undetectable on liver, hepatocytes, neural cells, muscle cells, and spermatozoa. Okay, and this as shown a while ago, this is the structure of the class 1 molecules. As you can see, the single chain that is being stabilized by beta 2 microglobulin, which is of course being expressed from chromosome number 15. So the glycoprotein dimers made up the 2 non-covalently linked polypeptides, and class 2 are primarily found on APCs. So these are examples of the APCs: B lymphocytes, monocytes, macrophages, dendritic cells, thymic epithelium, mast cells. So these are considered as the APCs, or the antigen presenting cells. So this is a structure of a class 2. So you can see that there are 2 chains here, and the class 2 molecules are the HLA-DP, HLA-DQ, HLA-DR. It is made up of 2 non-covalently bound polypeptides that are encoded in separate genes in the MHC. So let's talk about the role of class 1 molecules in the immune response. So as you can see here, class 1 MHC are actually important. It's actually important for peptide interactions, which means that the molecular peptide interactions involves the molecules process endogenous antigens. So together with this module, you'll be able to see an animation on how class 1 molecules work. So please do watch this animation so that you'll be able to understand what I'm telling you. what I'm talking about. So the peptides that can class 1 MHC interact to are supposed to be 8 to 11 amino acids in terms of length. So there should be about 8 to 11 amino acids, and the transporter associated with antigen processes include the top 1 and the top 2. So top refers actually to the antigenic peptides, and these are found in the endoplasmic reticulum. Okay, so please watch the animation that I will be uploading together with this particular module so that you'll be able to understand what I'm referring to. Okay, so class 2, on the other hand, participate in exogenous pathway of antigen presentation. So class 1 in endogenous pathway, class 2 in exogenous pathway. So molecules are synthesized in the endoplasmic reticulum and associated with a protein called the invariant antigen. So invariant antigen, okay, the I is cleaved to form a fragment known as the clip, which is exchanged for antigenic peptide. So the MHC peptide complex is transported to the cell surface. Okay, so again, I've been telling you that clinically, MHC is important in tissue transplant procedure because both of them can induce graft rejection, and they also play a role in the development of autoimmune disease. So as you can see here, these are examples of autoimmune diseases. So there are various examples. Okay, ankylosing spondylitis. If you have this, your spine will be inflamed, and it is associated with HLA-B27. Celiac disease. It is a type of autoimmune disease wherein you have malabsorption syndrome, and it is associated with diarrhea, weight loss, intolerance to gluten. Okay, so the chances of you getting celiac disease if you are expressing HLA-DQ2 about +3, HLA-DQ8, the strength of association is just +1. Okay, rheumatoid arthritis, it is the inflammation of multiple joints, and this is associated with HLA-DR4. Type 1 diabetes is wherein you have an increase in blood glucose because of the destruction of your cells capable of producing insulin, the Langerhans cells. So this one is also genetically predisposed to if you are expressing HLA-DQ8. Okay, strong association, and HLA. Okay, so there you have it. Chapter 2 lessons. So please continue to study very well. Okay, so God bless everyone.